Good morning, everyone, and welcome to this Hybrid Ag webinar. My name is Sarah Tinsley, and I'm excited to introduce our first webinar for this series, which will be looking at the stage of senescence. So through this series of webinars, we'll take you through the season looking into some of the technical aspects and scientific facts about soil health and plant nutrition. Hybrid Ag are on a quest to transform food for health, and that's our purpose. So we're continuing to curate research from the last 150 years worth of soil and plant nutrition research and data during our journey to discover exactly what it is that makes food healthy and how we can better grow nutrient-dense food. So I'll now introduce you to our panelists for today. So today we have Amos Rowe and Andrew Smith, and they're two of our research agronomists that work across a variety of horticultural crops with Hybrid Ag. And my name is Sarah Tinsley. I'm a soil scientist in the Hybrid Ag research and support team. So as we go through the webinar content today, if you do have any questions, please use the Q&A box at the bottom of the screen and we'll be able to answer them at the end of the session. But for now, Andrew, would you like to kick us off with our content for today?
Yes, thanks, Sarah. So good morning, all. So as Sarah's mentioning, today we're talking about senescence, which is the first stage of our webinar series. So today's webinar will be broken into 4 parts. First, we'll do a general overview of what senescence is. Then we'll look at the cell degradation process within the leaves, hormones that can influence the speed and extent of senescence, and finally, the nutrient requirements of a plant during this stage. Senescing plants is defined as age-dependent programmed degradation and degeneration process of cells leading to leaf abscission, or commonly known as leaf drop in plants. It is controlled and regulated by a variety of senescence-associated genes. There are 2 kinds of senescence, which is programmed and stochastic, which stochastic is actually random, which is when the tree actually breaks down further and loses all ability to control. The difference between these will be discussed a bit later. As leaf tissues are broken down during senescence, the plant is conserving nutrients and relocating sugars for different uses. In deciduous orchard trees or vines, this is when the leaves degrade and fall from the tree after post-harvest. These stored nutrients will also be put and used post-dormancy in the plant when it wakes up in spring. When the leaves are still on the plant before bloom, these are known as evergreen plants, for an example like an avocado tree. These evergreen leaves typically senesce the following year as flowering and fruit set are taking place rather than a one unified stage of senescence. These leaves go from being a source of energy through the photosynthesis process to a sink of energy through the senescence cycle. The appearance of these leaves during the senescence period is shown on the screen here within the orange box. They are seen to slowly experience colour changes from green through to yellows and oranges before becoming non-functional and then falling off the tree or vine in a process called leaf abscission, which is commonly known and referred to as leaf drop. So now we'll talk about the triggers for the senescence process. Senescence-associated genes can be activated due to a variety of stimuli, including 4 shown on the screen here. As a plant matures, this can trigger senescence. This is known as the age-dependent senescence. As lower daylight levels are experienced later in the growing season, this can help trigger senescence, especially as less and less light reaches the lower leaves of the plant. These leaves in the lower area will then begin to senesce, and this action will also trigger senescence in the upper leaves of the plant over time. Hormones including ethylene can help encourage senescence to begin, which will be discussed later in section 3. Finally, general oxidative stress, drought stress, and a variety of pathogenic infections can also bring forward senescence. Abscisic acid is a hormone produced during these stress periods in the plant, which links the stress and hormone triggers together. So the plant stress timing factors like these can influence the beginning of senescence differently, and this can be seen within the plants in the same block or in a particular row. So here we have a photo of 2 trees in the same block, which is in actually the same row. As you can see, the tree on the left has significantly more leaves, which is still nice and green and functioning, and shows little signs of senescence. However, the tree on the right has less leaves present, and those that are present are well on their way to losing their colour. So this is likely caused by different stress levels within the trees, which are pictured. So let's talk more now about the degradation of the leaf cells. I'll hand over to Amos.
Well, thank you, Andrew, and good morning, everyone. So to understand the cell and leaf degradation, let's first look at what we see on the outside. So during senescence, as we mentioned, the leaves change colour, die, and fall off the plant. And leaves initially have their green colour from a pigment called chlorophyll. And as we progress through the senescence cycle, this chlorophyll degrades, which causes the colour changes to the yellows, the oranges, the reds, those colours that we see through the senescence and leaf drop period. So these colours are due to underlying pigments such as carotenoid and anthocyanin pigments within the plant leaves, and we'll go into more detail on these pigments in part 5 of this series, of this webinar series. But it's what we want to understand is these pigments are always present, but they're not always visible until that chlorophyll is degraded, and then the other colours come to the fore. So chlorophyll degradation occurs due to a variety of senescence-associated genes becoming activated. And as the leaves fall off the plant, they take a significant amount of nutrients with them, and then the nutrients are then returned to the system through the soil. And we can actually aid this process by applying products that will assist and encourage that biological digestion of the leaf matter and the degradation of the leaves by the soil microfauna. So that process marks what we refer to as the beginning of programmed senescence. As Andrew referred to before, there's 2 elements of senescence, which are programmed senescence and stochastic senescence. And really the overarching concept here is that senescence begins as a plant-regulated process, which is controlled by the plant, and then it progresses to stochastic, which is basically random cell degradation later in the senescence cycle. So to understand how plants can store nutrients within their different carbohydrate storage sinks, we need to understand these 2 functions. And really the programmed senescence begins with the decreased photosynthetic activity within the leaves, which is triggered by several factors, including things like decreased light intensity, soil temperatures dropping, and things like degree days accumulation becoming lower. And as that photosynthetic activity decreases, the chloroplasts begin to degrade. And as they degrade and the plant begins to break these down, the plant needs to find a different way to source energy as opposed to photosynthesis. And there's a couple of enzymes that come into play here, being lipases and proteases. And these enzymes are responsible for splitting up of the stored carbohydrate molecules within the leaf into forms of nutrient that are more mobile in the plant once the photosynthetic activity is no longer present. And then with the breakdown of these energy sources, the overall stability of the leaves decreases to the point where the stochastic or random senescence takes over. And there's some information which talks about how the plasma membranes within the cells develop holes, increasing their instability even further, and then the cells begin to die randomly. But during this time period, a variety of different nutrients become mobile. So nutrients like iron, phosphate, zinc, nitrogen, molybdenum, and they can then be mobilized and stored within the plant in other plant storage sinks, such as the woody sections of the plant and that kind of thing. So we'll go into the mobilization of nutrients later in this webinar. But if we go to the next section, which is understanding the hormonal influences on the plant and how that impacts the senescence process. So Back to you, Andrew.
Yeah, thanks, Amos, and that's some great information there of what's actually taking place within the leaf at that particular time. So now when we look at encouraging hormones, senescence is significantly influenced by the hormonal balance within the plant. So we've divided them up into two groups for this presentation today. So those that can encourage senescence and those that can actually inhibit senescence. Hormones that encourage senescence include ethylene and abscisic acid. So during senescence, as the plant enters the stress state, it will release more abscisic acid and ethylene. Their impact can become more intense than the other major plant hormones like cytokinin and auxin. So abscisic acid, which is produced in the plant during the stress events, such as drought, also encourages the senescence process. So during senescence, often a reduction of light level can also stress the plant, resulting in the increased abscisic acid production. This will also inhibit shoot growth and has been shown to enhance the effect of ethylene on actual leaf drop. So we'll talk about ethylene now. So ethylene is commonly understood as a ripening hormone. However, it also is used in senescence. It has been shown to promote the natural detachment of plant parts, including leaves and also fruit. So this can activate a variety of senescence-associated genes. These genes, once activated, encourage the previously mentioned cell degradation process. And additionally, ethylene encourages the nucleic acid degradation. When these hormones are prevalent during the senescence process, they will continue to drive the senescence forward within the plant. So if you actually want to try and help accelerate the senescence process, you can also foliar apply abscisic acid type products, which will help induce that senescence process. So now onto the hormones that actually inhibit senescence. So the main ones to consider is the cytokinin. So cytokinin encourages the reproduction in plants, which we'll discuss more in part 2 of this webinar series, but it also can delay the senescence. So here we have an image from a study that's compared plants that actually overexpress cytokinin on the left with the wild type plants that express the normal amount of cytokinin on the right. So plants on the left actually show little signs of senescence with the nice lush green leaves, while the plants on the right show significant yellowing and leaf degradation. This same process can occur within the cytokinin dominant plants. So next we have auxin. So auxin encourages plant growth, which we will discuss in part 2 webinar of this particular series. So the auxin will delay senescence by inhibiting the degradation of the chloroplast within the leaf by maintaining the photosynthesis cycle within the plant. So another thing to consider is gibberellins will also generally delay senescence. However, the extent to which this will occur is dependent on the kinds of gibberellins actually present. Okay, so we'll actually go to the nutrient requirements at this time. Over to you, Amos.
Yeah, great, thanks, Andrew. So thinking about the key nutrients through this senescence period, to start with, in the senescence period, plants shouldn't ideally be exhibiting any major deficiencies, which we would aim to achieve by appropriate nutrient applications through the season at the other critical stages of plant growth or plant cycles. And the other way we can impact the nutrient levels at this point in time is through post-harvest applications, which are crucial in starting this process off on the right foot. When we think about applying senescence or applying nutrients at senescence, the main purpose is to ensure general leaf health and hormonal balance, which is important for the reasons that Andrew's just touched on. And then as the leaves degrade and the photosynthetic activity decreases, the nutrients will be stored in the other plant parts, like the woody tissues and the buds. And in some cases, they can actually be stored in the form that they're applied in. So taking the nitrogen form of urea, for example, applying a urea form of nitrogen at this stage can mean that the nitrogen actually stays in the amine form as it's not broken down by photosynthesis. And that's important for a couple of reasons. So the amine form has high freezing point and can provide a certain level of frost resistance. And the other aspect of it is that it can provide a very available protein source to the plant as the plant is waking up or in early stages of bud growth in the following season. So another nutrient to think about is magnesium. And as you know, magnesium forms part of the chlorophyll molecule and is quite important for energy transfer within the plant, as well as the maintenance of cytokinin molecules. So applying magnesium at this time of year provides a key element for energy transfer that the plant isn't able to easily access when there's no photosynthesis in the new season. With zinc, one of the key things that we think about with zinc is that zinc has been shown to provide energy which helps us to establish significantly larger spur leaves at bud break. And these spur leaves are the first place that the plant begins to draw nutrients from when it begins to encounter nutrient deficiencies later in the season. And also zinc is important for strong pollen structures, stronger seed setting, and things like the formation of cell wall membranes. And there's also a component there with resistance to pathogens and helping to form proteins. So all of these nutrients are important, and they can also assist in speeding up the leaf drop process. And they're commonly used, particularly urea, through the industry for that very purpose. Finally, abscisic acid, technically not a nutrient, but worth a mention because there's been studies that have been done that show that applications of abscisic acid products can accelerate leaf drop, similar to what we see with the applications of urea and magnesium and zinc. So senescence applications, we can also tailor them to cover or address any nutrients that are known to be an issue at this point in time to bring optimal balance into the plant through that dormancy period. So I'll just hand you back over to Andrew for some grower take-homes.
Thanks, Amos. That's great. So yeah, just to sum up today, based around some grower take-homes. So we'll actually look at what we've discussed today. So senescence is when leaves degrade and become non-functional, falling from the plant. So this can be triggered by light intensity shifts, hormones, or other stress sources. It is regulated by a variety of factors, including some genetic as well as hormonal balances. So leaves initially degrade with programmed senescence, but as cell degradation becomes random, the overall plant stability is lost. So when we move and talk about hormones, so they can both encourage or they can inhibit the senescence cycle, just depending on what's present at a particular time. And then we look to the nutrient applications before senescence should really be matched to the deficiencies of the plant post-harvest to achieve a balanced nutrient storage during that senescence cycle. So the other thing to probably consider is soil testing to actually assess the nutrient levels will also be crucial to achieve the plant's best genetic potential throughout the next growing season and should be reviewed annually post-harvest. So with that, I'll hand back to Sarah, and I'd like to thank her for all her input in today's webinar. It's been great. So thanks, Sarah, over to you.
All right, thanks for that, Andrew. And thank you again to everyone for joining us in our webinar this morning. So in our next webinar, we're going to be following on from today's discussion, looking at the stages of bloom and pollination and of cell division. So that will be happening sometime in August, which we'll confirm a time and a date for. So keep an eye on our social media for details about that. Additionally, this webinar will be uploaded to our YouTube channel. So if you would like to review this content again, we'll be able to send you a link for that video when it's uploaded. And finally, if you have any feedback from this webinar that would help us improve in the future, then we'd love to hear from you. So please contact us at either support@hybridag.com.au or give us a ring on 5722 7555. So the questions from this webinar will be covered in a document that will be emailed out to everyone. Now let me have a look. So for now, we just have the one question. So Andrew, would you like to tackle that one?
Okay, all right. So we've got a question here, which is of interest. So do sulfate-based trace elements work during post-harvest process? So probably just quickly without going into great depth, sulfate-based trace elements have probably been consistently used throughout industries many years. Probably the key thing here is we want to make sure that the elements are going into the leaf. So I'd probably prefer to see a chelated type product used because we know that will definitely hit the target and get to the leaf because we'll make sure that the nutrients are stored within the cells. So the tree, when it starts its senescence cycle, has actually absorbed all that post-harvest goodness that you put out. So that's probably it from that particular question. Thanks, Sarah.
All right, great. Thanks for that, Andrew. So if anybody hasn't asked a question today, but you would like more information, please don't hesitate to drop us an email or give us a call. So those details are on the screen just again. All of the questions and answers from this event will also be available through our certified partners, who are David Morello at Morello Fair, Noel Jones at Sahara and Elders, Craig Swansbury at Fruit Doctors, and Graham McNeil at Chippy Crop. And finally, we'd like to thank all of the technical experts from around our industry that help bring you these events. So we'll see you next time, and don't forget, happy soils make healthy souls. Goodbye.